Yes. So good evening to everyone. So welcome to day three session on biological databases. And we today we'll be discussing on data file formats, metabolic pathways, motif and domain databases, and even we discuss on network databases. Before proceeding on to day three session on biological databases, this is an extension of day two session topics. Okay. We recap the last session topics. Yeah, can we recap the last session topics? So, as usual, what are the last session topics we have discussed? So, come with your answers.
Sir, good evening, sir.
Good evening.
Yesterday classes the biological databases, especially in the IEDP, that is for the immune-free epitope databases, and you have to explain in the T cell and B cell. Then molecular sequences, what is gene and what is genome, what is in the coding. Then you have to explain in the many of the softwares like the proteomics pasta, and then TTC, and then protein sequence, 3D structure, enzyme compounds and pathways and complexes, and what is the TDBJ and KEGG. So you have to explain the more software and on yesterday's.
Good, very good, Shobana. You did a very great job. So, I think from top to bottom, she has explained everything. How about the others? Can I get an answer from even from the other participants? Excellent. Even the day two session, day one session, I think she also has recapped everything. How about the others? Because we also find participants from other colleges also, from 16 more institutes. Yeah, we discussed on DNA Data Bank of Japan. Very good. Like this, at least whatever you acquire, it's there in your mind, whatever is there in your RAM, you can discuss. And your participants, one thing you should understand, the audio quality is based on the internet frequency. Here I have 5 star, 5 points internet strength. How about you? Sometimes we find the frequency in wave bandwidth, isn't it? So one thing you can do, you can whatever I send the recorded videos, audios, you just have to watch the videos. And one a few participants have mentioned that taking notes while providing the demo is difficult. Yes, it's very, very difficult. See, I am taking lot of concepts in a short period of time. So only thing is that you can watch the video for the first time. And if you play for the second time, you can start taking notes. There is no chance to take notes because I'm taking in stretch. It's a continuous kind of a session. Audio is not clear still. For Gauri, is it not clear? How about the others? How about the others? So for one participant, still the audio is not clear. I want to know whether really audio is not clear or not. Is audio clear to everyone up to present? Yes. See. Yes. See, it depends, Gauri, and even for the other participants, it depends on your internet frequency. To try to have a kind of a good kind of a frequency internet. Okay, in your place, I think you have some problems. I suggest you to watch the recorded videos if you have any problems, or you can see where you find the internet frequency band. Okay, so just watch the video. Okay, and then second time you can start taking notes. Okay, this is my kind of a feedback for you. And see, still I'm admitting the participants. So this is a very easier way of accepting the Google invitation, isn't it? I just invited in the Google Calendar. See, these are the participants who are yet to accept my Google invite. So Harish Kumaran, Kalai Shelvi, Nayak Gauri, Angel Priya Singh, Riyas. If you accept, then I'll be getting this tick mark. Okay. And see two more participants. Just now I got from her that she is going to again request. So if she requests, then I can once again admit her through the Google Meet. Now it's not possible. I have to close and I have to admit once again. Okay. So now these are the participants who are yet to accept the Google invite. If you accept the Google invite, there is no need to disturb others and also me. Okay. So try to follow, find the invitation, or you just message me in WhatsApp or in my mail so that I can re-invite once more. Okay. So let me come across because I'm getting some audio quality is not clear. Then this is the only way. Okay. Why we have to talk unnecessary, isn't it? That's what one feedback I got from one participant. Okay, let me go ahead to not even disturb that kind of a participant. Today we'll be discussing on biological databases, data, and I should appreciate the participants who have given good answers during this time. So I got answers from Vijaya Lakshmi, Vijay, and then Sandhya Gopi that we discussed on different biological databases like GenBank, KEGG, SCOPE. SCOPE was actually, we have discussed like SCOP. So then I think I have to ask few questions. Whatever you understand, you have to drop the answers. Can someone expand SCOPE? Because this is what we see in final day MCQ. Expand SCOP. SCOP is a secondary database, so we find protein information, protein domains. Then what is SCOP? So from your questions or from your answers, I'm just asking you. And similarly, expand KEGG. What is KEGG?
Sir, SCOP, structural classification of protein,
Very good. Structure classification of protein is perfect answer. How about KEGG?
is especially used for identify
Sir,
pathways, disease networks. Sir.
Ah, you're right. Okay. So the purpose is right. The purpose of KEGG is right. Actually, Kayeto, yeah, yeah. So some more.
genome.
Ah, yes. Again, this
Sir,
same participant is.
TBD is highest level of functional utilities of biological system.
You are right, madam. So all the participants, those who interacted, answered well. Now let me go to something new, whatever we are not discussed. Okay, good. So now I take you to that, and if you face any problems, okay, in any understanding any concepts, even you can message in the Google WhatsApp group. I mean, sorry, the WhatsApp group or even the private WhatsApp, so that I can try to explain you. Okay, because these are something you are studying and you are getting some kind of a doubts while you are preparing. Now let me take you to the today's session contents. Today we'll be discussing on data file formats. Okay, because we know the so many public domain databases in primary database, secondary database. We have types of databases we have discussed on yesterday, isn't it? We also discussed composite database. I have taken an example of ProSite as for composite database. And for primary database, I provided example like NCPA GenBank, NCPA GenPep, and like this DDBJ. This comes under primary sequence databases. And secondary, we come across COP and CAN. How about the file formats in these databases? We see in different databases in this session, and that we discuss on different biological pathway databases. We have just discussed on KEGG. How about the other databases? And how about the motif database, domain database, genomic database, cancer? In genomic, we have discussed NCBA genome. How about the other databases? What are the different cancer databases available, and similarly the gene expression, gene enrichment. And this is one such a database I have developed in my lab. Not just me, myself and the two PG scholars have developed this kind of adjuvant and immunoanalytical database. Okay. uh, but I thank all the participants who are even in the feedback you have mentioned that it's very clearly understanding. So thank you. And this is an opportunity you can pursue PhD under my guidance. So this will be under Anna University. Uh, Now about the data file formats. Bioinformatics database identifiers. Um, so these are the common identifiers for bioinformatics data: locus name, accession numbers, gene info ID, and PubMed ID. So if you see the locus names, so instead of searching human beta-globin, we can also provide keyword like HUMPB. Okay, and so generally we can give three letters of organism followed by code for gene. You know, this is for NCBA GenBank. In default, in NCBI GenBank, that is NCBI nucleotide. If you go to the NCBI URL and if you select the nucleotide, we get a GenBank format, isn't it? So if you don't know about a particular GenBank accession number, how come we can able to get the sequence of a DNA or RNA? So you have to type a keyword. The keyword can be a protein name or even an organism name, or even it can give an example like this. For a human beta globin, collectively give this small keyword. Like this insulin, INS. Like this, we can give hemoglobin, HGB. So something, whatever you know, some three-letter words also you can give. Okay, and you can also check if whether you're getting or not, or else give the full name. And second database identifier we find is the accession number. Here, if it is nucleotide, it is GenBank. And if it is even protein, we tell like a GenBank accession number, but the type of database is GenPept for protein. That's the only difference. Can be a nucleotide or protein, we have GenBank accession number for it. The GenBank accession number is like this, alphanumeric. Okay, so alphabet followed by how many? 1, 2, 3, 4, 5, 6, isn't it? But for a few nucleotides, we also have 7, 5. So it varies actually. It's not consistent. Okay, but one thing you should remember is it is an alphanumeric letters. And what is this point one? This is a version number. Okay, so this is not fixed actually. 567 number is there, and for a few uh, sequences we find this version number point one. And the third database identifier we see is gen info id. Like GenBank accession number, the gen info id is followed with gi followed by a digit number. So this is consistent here, gen info id. So whatever I'm telling, like a GenBank accession number, gen info id, locus name, these are identifiers for sequences present in NCBA. Can be a DNA, RNA, as well as for proteins. And if you see the fourth database identifier, this is PubMed ID. The PubMed ID is generally provided for the biotech literatures, but also the PubMed ID can be provided for the sequences. The PubMed ID is also an 8-digit number. Okay, so these are some of the database identifiers, like how I can identify roll number, registration number, name with initials for the student. Similarly, for the sequences, we can able to identify through this database identifiers. And if you see the bioinformatics database formats, okay, we can see the universal format is the FASTA, and we also have GenBank. So the FASTA and GenBank are available in NCBI. And in Uniprot, we have a format called SwissProt. Okay, I told you in Uniprot is a major uh, database where in Uniprot you have a database called KB, Uniprot KB. The Uniprot KB is is actually here you can find two types of databases. One is SwissProt, the other is TREMPL. So these are the two databases linked with the Uniprot KB. And SwissProt is the format name for a sequence. And ASN.1, this is also in a universal format, present in most of the databases. XML, I think you already heard about this, extensible markup language. Okay, this is also a format for sequence. Now let us see the database format of FASTA. In FASTA, we have this greater symbol. Actually, in Unix, we call this as filter command. This is a pipeline actually. No, not filter. This is a pipeline, Unix pipeline, greater symbol. So you are giving some kind of content. So whatever identifiers I give, the first identifier is gene info ID. And the next what we find is the GenBank accession number. See here, only we have 5 numericals followed by version. It depends, okay, based on the sequence. So this is not consistent. And whatever we find, this is the locus name. The locus name can be an abbreviated word, and this can be also be a species name, and it can also be a protein name. The protein name is breast cancer antigen 1. That's what BRCA1, and the corresponding sequence is mRNA. I mentioned you on yesterday itself, even for mRNA, we won't find U, but we find even T for instead of U for mRNA. This is how a bioinformatics data looks like in any public domain biological databases. Now let me go to the Next database format, GenBank. This is another format we can find in NCBA itself, but we find some data which is pertaining to the species. So if you whatever you find in organism, okay, that's a species, and if you want to see what exactly it is, we can able to see it in the definition. In the definition, we can able to see mus musculus. What is mus musculus? I think everyone know it's mice. So mice, breast cancer antigen 1 mRNA, and this is complete CDS, complement determining site. Okay, this is a complete sequence. That's what you can understand. Complete mRNA sequence, and mus musculus is the species, and this is the taxonomy. Other than that, we can also find the reference authors. Who are the authors who have posted the sequence, and who are the authors who posted in a journal paper. So if they posted in journal paper, we can also get this journal name. Okay, this is the journal name, and we can find even the volume number, issue number, etc. And this is the identifier for downloading the journal paper. So we can get two different kind of an authors. One author is who has posted the sequence in the NCBA, and the second author, the part of the text only we can able to see. If you drag down in the NCBA GenBank format, you can also see two different authors. One author is who has posted it in the journal. The other a set of authors who has posted the sequence in the website. Okay, that is in the NCBA GenBank. And the third database format is the SwissProt. In SwissProt, like whatever you find in UniProt, okay, so you can also have an exclusive database also called SwissProt. I told you in UniProt KB we have SwissProt and TREMBL. Other than that, there was also an exclusive database called SwissProt. If you find a sequence in the SwissProt, we can able to find like this, like however you find that ELF GenBank format. Okay, we find some information like the type of protein, the type of molecular sequence, and type of the species, and the taxonomy, PubMed ID. Okay, and the fourth one, what we are seeing is the XML. This is a database format, another database format. XML stands for Extensible Markup Language. This is actually a database language. This is a semi-structured database language. We also find the entire program in the form of tags. But in HTML, whatever tags we have that are predefined, that has predefined functions. Okay, for example, in HTML, okay, if you type something like bioinfo, okay, inside a bold tag, this is a bold tag, this is an open tag, this we call it as an open tag, and this is what we call it as a closed tag. The closed tag has this slash. So whatever you open, you have to close with the same tag through this closed tag. So in HTML, okay, if you type this code, okay, we display only bioinfo. But whatever we display in bioinfo, that is in bold context. Like see here, these two lines are in bold context when compared to the content what we find in the below, isn't it? So if you want to make a bold, or if you want to make it as italicized, use I /I. So the tag is nothing but a less than or greater symbol. So whatever text you enclose, you get a corresponding function in the HTML. I want to mean that HTML is a web-based language where we can able to design the web pages using the tags, which has a predefined function. Okay, but whereas XML is a database language, we use user-defined tags. We can have any name. I can give something like SSN, okay, and then I can close the SSN. So in SSN college, if I want to have the faculty database, I can open the faculty, and then I can start with the department bio, and I can give the list of faculty names inside, and then close bio, and then you close the faculty. Now, if you want to feed this student, you can again start with the student tag and then give the data. So whatever we are going to give inside, these are the data. See, in XML, how we started. Initially, I started with XML tag. And then even while closing the program, I have to close with XML. Inside that, I can give some user-defined tags. I can give whatever I give, SSN, bio, C. These are my user-defined. whatever I feel more understanding for me, that I am giving inside. Okay. Similarly, we can give like GB, that is GenBank set, GenBank sequence, GenBank locus, and in the locus, I am giving this kind of a data, whatever you have already seen in GenBank format, whatever we have seen the length in the GenBank format. Okay, so like this, you can see the XML language, but what I want to mean is we also have a database. I mean, data in the form of .xml also. Now we discuss on biological pathway databases. Till now, we have completed about data formats, and even yesterday, I believe I have shown you how to download the FASTA format using NCBA GenBank and in Uniprot FASTA, Uniprot KB, how to download the FASTA format, isn't it? So whatever formats I showed, I can also show you when I go to the web. Now let me complete something in PPT. Now let me take you to this pathway databases. I believe even I have taken you to this. Okay, but I didn't take you to the hepatocellular carcinoma, but I have taken you to the asthma and glycolysis. In hepatocellular carcinoma, I am actually doing research in hepatocellular carcinoma for more than 7 to 8 years, I believe. So more than 8 years, I worked in this hepatocellular carcinoma. I worked two funded projects in the hepatocellular carcinoma. So here, whatever we see, uh, we can able to see nearly 8 important pathways in hepatocellular carcinoma. But myself, I just worked a novel kind of a target, and I worked on a novel kind of a pathway. But most of the important pathways you can able to see for a disease. Similarly, if you are working on a particular disease, you can see this. But most of the research are already saturated. So one has to go with a strong literature review for doing a novelty research. Now let me take you to the next database in pathways. So first, what I showed you is the cake pathway. The next I am going to show is string. The string is the largest database for protein-protein interaction networks. When we need these kind of a data, protein-protein interaction networks, if you go with a proteome study, okay, yesterday also I told you what is genome, how the human genome is constructed. Okay, I told you the shape of a human genome, and I mentioned you what is the composition of human genome. Remember? Now, when I'm explaining you about the genome, I also mentioned about the proteome, a complete set of proteins in an organism. That's what proteome I mentioned, isn't it? We also should have a knowledge on the protein-protein interaction networks in that. So this is very important to come up with a precision medicine for any type of disease. Okay. So some 15 years before, 20 years before, we don't have these kind of a networks. But this is something, a contemporary research happening at present era, that is in bioinformatics era. If you see this protein-protein interaction networks, we have around 40,094 organisms and 67.6 million proteins. And the types of, the length of interactions is more than 20 billion interactions. So that's what I told. This is the largest protein-protein interaction database. What is this interaction 20 billion? What is this 67.6? So to know about this, if I go to the web, then you can understand better. So these are some of the functional enrichments. I don't think you can understand. Now better I go to the web and then I come to the PPT. So to understand about this, I have to take you to the web. So let us take you the web. So first we can see NCBA once again. In NCBA, no, we have seen the FASTA format. But did you see that GenPep format? For GenPep format, I'm taking it a protein followed by insulin. Go to any kind of GenPep format. Now I'm taking for Octodon. No thanks. No need of survey. So this is how a GenBank format looks like. So the first set of authors posted the sequence and second authors are there. No, we have only one set of authors. But in few sequences, you can find two sets of authors. If they publish a journal paper, then we can find. So here the journal paper authors alone are shown. Most probably these are the authors who posted the sequence. Now let me take you to the next thing, string. So string TV. If you type string TV, it take you to the string-tv.org. So this is a functional protein association networks. So here, these are the updated information. Each and every day they update the protein-protein interaction networks. To search any data, click search. Now you have to type any protein name inside. So I can give insulin or I can give something new, trap1. You know the trap1 is tumor necrosis receptor associated protein 1. This is my protein of interest I'm working in my lab. So if I type a protein name, so defaultly it's asking for whether you want to search for the Homo sapiens. Default selected is Homo sapiens, but we also have 261 different species. Mus musculus for mice, Drosophila, fruit fly. We also have other kind of trap1 associated protein in human. But if you want to go with the trap1 of human, just click continue. If you click continue, then we are getting this kind of a network. See, this is actually what kind of a network? Protein-protein interaction network. This is our hub protein. This is our hub protein trap1, and this protein is having interaction with the top 10 important proteins.
Sir, excuse me, sir.
Uh,
PPT is not visible, sir.
PPT is not visible. Is that
Yeah.
website is not visible to you? Website? I am.
No, sir, everything is not visible, sir.
Oh, yeah, yeah. Okay. How about the others? It is visible, sir. See,
It is
actually,
visible, sir.
good. Thank you. Actually, the problem is with the internet. If your internet bandwidth is low, you can't able to see. The only thing you can manage is after sending you the recorded video, you can watch it multiple times. Then you can have a knowledge of it. Okay. So try to have a good frequency. That's a problem actually for the other participants also, not only for you. Those who just mentioned me in the feedback that even the video quality is poor. Okay, now let me take you to this website. Good, but thank you for interacting. Okay, now let me explain you further. So trap1, actually is interacting with 10 important proteins. Okay, uh, if you want to know about a particular protein like HSP9A, A9, so click this. If you click this, you will be getting this information. Okay, like if you want to click any specific kind of routine which is present in the form of a circle, you can get this information, detailed information. So you also have the identifiers. If you go to the identifiers, you get so few more information on that. Okay, the structure, PDB ID of the particular protein, whatever is interacting with the trap1, everything is available here. But if I'm not focusing on the 10 proteins, but if you want to get the top 100 proteins or top 50 proteins interacting with my hub protein trap1, how to make it? This I can able to make it if I go to the settings. Go to the settings. In the settings, okay, you have first cell no more than 10 interactors. But if I want to go with the around 50 interactors, okay, in the first shell you have to select like no more than 50 interactors. So then what you will be getting is you'll be getting the 50 top interacting proteins. Now how to get this? So after selecting this parameter, click update. There is a button called update. See, these are the 50 top 50 interacting proteins. This is something important. If you go with the gene expression, if you want to go with the precision medicine, I mean, if you want to precisely target a particular patient who is suffering from a cancer, of course there's a cancer target protein. Okay, so if you want to target a specific cancer target protein, don't limit up to even a few interacting proteins, increase to a larger number. Then you go with the gene enrichments. Okay, then only you can identify what is the problem and what is the important target interact. I mean, what are the important proteins which interact with our hub protein. Don't just target with the trap1 protein. Also target the other important proteins which are interacting more. It can be a positive regulating protein or it can be a negative regulating protein. with tryptophan expression. But our objective is to go with multiple and then screen based on gene enrichment. Okay, so all these things you can learn in the subsequent classes or even in the other kind of internship courses where I take you in deep. Okay, now this is something about how to find a top 50 interacting proteins for a particular protein, whatever we have searched. How this data, how we are getting this kind of a data, we are getting from this, that is, actually this, or we got from various techniques. The techniques I can show you, that the co-purification, co-crystallization, yeast to hybrid, genetic interactions. So these are the wet lab techniques, experiments they have used to come up with these kind of protein-protein interaction data. Also, whatever information available in protein databases, metabolic pathway databases, even these are helpful to fetch in the form of this protein-protein interaction networks. Okay, co-expression, co-occurrence, co-occurrence, fusion, neighborhood. These are some of the analysis they have done. And there is one button called analysis. In analysis, okay, in analysis, we have functional enrichments. Under the functional enrichment, we have biological process, molecular function, cellular component. So if you were specifically focusing on a particular type of cancer, okay, for example, I'm going to show you, like, if you're focusing on a particular organ, okay, click this. For example, if you're focusing on liver cancer, I select liver. If I select liver, not this, you get an orange thing. For a liver, you get an orange. And if you want to focus on a particular pathway, can be a TNF alpha signaling pathway, select this. Now we're getting violet. So one particular organ and one particular pathway, okay, orange and blue, that violet you can see here now. Orange stands for the most important target proteins for liver, and the violet stands for the proteins which contribute to the TNF pathway. So if you see only these two proteins are, I mean, the tryptophan is there obviously. Along with the tryptophan, the most interacting protein which shows, which is screened from these two filters, is CREB. So this is the most important target protein, and also CDC37. Like this, we can able to find in protein-protein interaction networks. So whatever data I just screened, the data is from the string DB. And if you want to download this information, you can able to download the information through exports. In exports, we have a tablet text output. Tablet text output. 1, 2, 3, 4, 5. Okay, the fifth one. It's in TSV format. TSV is tabulated separated value. So this kind of downloadable file can be opened in Excel. If you want to explore the data, you can able to explore it in Excel. We have a folder called Bioinfo25 in my desktop. So I'm going to store this kind of a data at present. So whatever protein-protein interaction data I have downloaded from string TV, if I want to open, you can able to open through Excel. So better I can open Excel and just, so first I open an Excel. After opening an Excel, what I'm going to do? I'm going to drag it and drop it. Yes, now see, whatever I dragged and dropped, we got this information. So this is a protein-protein interaction network. Okay, these are the coordinates of the files. So AH is A1. Okay, this is a protein interacting with another node 2, PPP. And how about the information of the interaction? This AHSA1 is also interacting with another protein called PSMC4. Not only the tryptophan is interacting with all these proteins, even the proteins, whatever they find, that remaining 49 proteins are also interacting with other proteins. If you see HSA1 is interacting with around 22 proteins, 23 proteins. If you ignore the first row, it is this HSA7 is interacting with 23 proteins, but TRAF1 is interacting with 50 proteins, whatever we can see in this Excel sheet. So like this, we can find the information of the protein-protein interactions. But if I want to open the same, okay, in the visualizer, we have a visualizer called Cytoscape. So one should install this Cytoscape. The Cytoscape is a freeware. To open a protein-protein interaction networks, okay, open the Cytoscape visualizer, and inside the Cytoscape visualizer, you can able to open the networks. Whatever we can able to see, in this, sorry, yeah, whatever you can able to see here, the same thing even we can able to see in this visualizer. So to download the Cytoscape, okay, better type Cytoscape in Google. So defaultly, it takes you to the cytoscape.org. So in this platform, you download the recent version 3.10.3. Okay, just go to the home page of the website. Don't go somewhere else. Okay, now let me take you to the PPT and continue further. So more about the Cytoscape we'll be seeing in the subsequent classes, subsequent sessions. Now let me take you further to the functional enrichments. Okay, this is something pertaining to the string database. I told you, while you want to filter the particular proteins in the network, we can select any of this. Initially, I selected an organ, and then I selected a particular pathway to select which are the most important proteins. I found only 2 important proteins are connecting with the trap in the enrichments, isn't it? So I selected the liver, and here, I also selected one more thing called hepatocellular carcinoma. That is a primary liver cancer. So violet and orange, these are the most important targets. And the most important proteins I found is these are the proteins. These are the top important proteins. This is the first most important protein for hepatocellular carcinoma that we can able to find from this. And this is another database called compound protein interaction database. We call it a stitch. So earlier in string DB, we can able to find only the protein-protein interaction. But whereas in stitch database, we can able to see a compound. Vithaferin A is a plant compound from ashwagandha, Withania somnifera. So you can see how a plant compound is interacting with the proteins. We can also see drug, indomethacin, celecoxib, this is another drug, how these are interacting with the proteins. So one thing you have to remember that here it is not just limited to the protein-protein interactions. You can also see the protein-protein interactions in stage. Other than the protein-protein, we can also see the compound-protein interactions. And this is a third database, protein-protein interactions, FunCoop. So it looks very funny. So I can take you to the website also. Here also, you can see the protein-protein interactions. We can also able to filter by tissue, filter by pathway, and then we can see such a kind of networks. So there are actually 2 different portals to deposit the sequences. One is Sequin, the other is Pancake. If you want to deposit a new nucleotide sequence, can be a DNA or RNA, better go with this standalone software. Any scientist, any student or a scholar, if they identified a gene, if they sequenced a gene, they can deposit the sequence in NCB. Anyone can deposit. So if you're depositing, either use Sequin or Pancake. That's what I want to mean. And if you see this difference between these 2 tools, Sequin is standalone. That is, it's an offline. You have to download and install Sequin. After downloading and installing, from the desktop, you open it and then try to submit the sequence where it guides you to the multiple steps. Nearly 4 to 5 steps over there. You just use that 5 to 5 steps, around 5 steps, and then you can try to get it. So 5 to 7 steps, I believe. Okay, however it is, you have to follow the steps in sequence. Okay, and then so what you find, while submitting the sequence, it asks what are the name, who are the persons who come up with this kind of a sequence. So we have to feed the names, and we have to feed some more information, journal information, like whatever pertaining to the NCPA GenPEP, in GenBank format. Okay, so this is a standalone. Then how about the Bankit? Bankit is an online submission. Bankit is an online submission. Okay, that is, it is a web based. That's the major difference between Sequin and Bankit. And in Sequin, we can able to submit complex submissions containing long sequences. But Bankit, what is small sequences? No complex, simple sequences. Simple sequences can be submitted. So this is the major difference between Sequin and Bankit. Now next about Cath, about secondary database. I told you on yesterday, CAT is classification, architecture, topology, homology. Okay, so what is class actually? This is actually overall secondary structure content of the domain. In a protein, we have nearly 5 levels of protein structure. So if you go to the third year or fourth year, you can study the protein science, where you can find in levels of protein structure as primary, secondary, so like primary, secondary, and tertiary and quaternary. Okay, so in between tertiary and quaternary, we also have super secondary. Super secondary. Okay, so totally we can tell we have 5 levels of protein structure. So all the proteins are classified in these levels of protein structure. If so, if you see this secondary database like CATH, okay, these proteins are classified in this architecture. Okay, so I can see the detailed description of this. One can go to the CAT database. Yesterday, I believe I've shown you directly to the CAT database. Go to that URL and you find this information. And SCOP is a structural classification of proteins. This is another secondary database where instead of classifying it in 4 stages, 4 classes, here we are classifying it into 5 classes. Sorry, 7 classes. Okay, we classify the proteins in 7 classes. Like the first is a class, and next is the fold, superfamily, family, protein domain, species, and seventh is the domain. Okay, this is what structural classification of protein. Now let me take you to the genome specific databases. In genome specific databases, we already discussed on how to find a genome from NCBA. Similarly, one can able to find in THER. The THER is the Institute of Genome Research. Actually, these are the largest genome websites. Okay, these are the largest genome websites. However, in the 14th session, we'll be discussing more on the genomics and proteomics. I'll be taking you the types of genomes databases, and I will be taking you with each and every genome and proteome databases in detail. And CAKE, we already discussed Kyoto Encyclopedia of Genes and Genomes. We can also find the gene information in this CAKE, like how we find in NCBA. We have plenty of genome specific databases. Ashwagandha is belonging to a family called Solanaceae. Ashwagandha. Ashwagandha you find in tea, isn't it? In the masala tea, you can find this ashwagandha. Why we use ashwagandha, to reduce the stress. It belongs to the Solanaceae family. You know what is Solanaceae? Solanaceae is nothing but the tomato family. Even tomato belongs to the Solanaceae. Whatever you find like berries, tomato, red berries, even the ashwagandha fruits looks like a tomato berry. So if you see, for ashwagandha, we have a separate genome website. For a tomato, we have a separate website. We have so many kind of breeds in tomato also, isn't it? And if you see this, even in ashwagandha also, we have the different strains. And we also have an exclusive Solanaceae genome website. Okay. So not only you find the common genome websites where you can find the plants and animal species inside, including insects, worms, everything you can find in NCBI genome, but we also have exclusive databases for the plants, also for the family of plants. Like this, we have plenty of genome databases. Thousands of genome databases are available in the world. NHRI, like the National Human Genome Research Institute, the NCB is maintained by National Institute of Health. The National Institute of Health is also governing this kind of genome website. So this is maintained by National. This is the division of NIH which is taking care of maintaining the genome database. So one has to go to the genome.gov. And we also have a National Institute of Plant Genome Research. Okay, it's in our country. So if you want to go to this genome information, these genomes or indigenously developed by our nation people. Okay, there are so many genomes deposited, plant genomes deposited. One can go to the website and look through the information of that. So we can also find the cancer genomes from mycancergenome.org. This is the URL. Go to this URL, go to any specific disease, okay, any specific cancer, and then you can find the types of genes responsible and what gene, how much percentage. In hepatocellular carcinoma, TP53, this is the most important gene. Nearly 23% of the patients who infected with hepatocellular carcinoma, you can find this TP53 gene. So this is the number one. Okay, like this you can check for any types of cancer. So somewhere you can find the TP53. If you click this plus thing, you can find the percentage of occurrence in humans. Got it? Now let me take you to the another kind of a database. We also have exclusive databases for a specific type of cancer. One thing is a database for hepatocellular carcinoma, DBMHCC, oncodb.hcc. This is another exclusive database. This is from Taiwan. Okay, like this, each and every country also have an exclusive kind of database, even for specific types of cancer. Cancer liver. This from an Indian database. This is a cancer gene expression database. This is developed by Raghava professor, okay, from IIT Delhi. He has developed so many tools and databases, like my mentor, Professor Michael Cromiha from IIT Madras. Okay, so these two scientists have explored a lot in bioinformatics. C Bioportal, this is a public domain database for cancer. Okay, you find plenty of information in this database, like how you find NCBI, GenBank for nucleotide. If you want to find more information on the cancer, one can also go to this cbioportal.org. OMIM. You remember yesterday I was mentioning you about this online Mendelian inheritance in man. If you're working on hereditary disorders, yeah, I think even in the feedback, I got two types of constructive feedback. Like the participant was asking, like, "Sir, try to provide me more information on genetic information, genes information." So see here, this is what OMA. So the gene information you can find only in OMA and not in all the databases. Okay. So if you're working on gene related, go to this, explore more, work more on this. Okay. Like if you're working on animals, better go to the OMIA. And few participants also ask me, sir, try to give me something related to plant-based drugs. So for this only in the WhatsApp group, I think you have seen on today morning, I have posted the YouTube links. So the YouTube links, whatever I pasted is I delivered to the reputed companies, bioinformatics companies. So go to that kind of websites also. Okay, the YouTube videos, whatever I sent, which will be helpful for you. Like practically, how can we can understand all this? No, I can't able to deliver everything in the 14-day session. So one day sending you some other additional YouTube URLs which can help you. Now, this is another database called Genomic Data Commons Data Portal. Okay, so that is to go get inside the genomic data commons. That is, you know, in US also you can find patients from hepatocellular carcinoma. In India also you can find patients from hepatocellular carcinoma. Then how can we can distinguish one origin from the another origin? Actually the patients have a different set of genes. Not for all the, I mean, the patients from India have can be diagnosed with a different gene for a specific cancer, and patient from here can diagnose with another gene for the same cancer. So then one has to explore on the other set of genes, especially they have to focus on even the HLA phenotypes. The HLA phenotypes vary from person to person, like how we each and every person can vary with the fingerprint. Similarly, we can able to see the HLA genes vary from person to person, including for siblings. Even if you find twins, for twins, the HLA genes alone vary. But HLA, no, these are immune genes, human leukocyte antigen genes. But if you see genes of myoglobin, hemoglobin, muscle protein, blood protein, this looks similar for any country people. Okay, but what is this HLA genes vary with respect to person. So if you want to focus on more about the this kind of a genes and the most common thing we can find in GDC, and GEO is the gene expression omnibus database. That is, if we want to focus on a particular disease gene expression, can be SARS-CoV-2 expression or can be a cancer gene expression. So better go to this GEO. This is also available in NCBA. Gene cards, this is also most important database. If you are working on genes, better go to this gene cards. If you're working on genetics, go to OMIM. This is the largest database for gene sequences. You get almost all the genes information here. So for the hepatocellular carcinoma, 8,100 genes are available. 8,100 genes are responsible for causing this primary liver cancer. All the genes information are available. If you see this TP53, if you click this plus, you get a detailed annotation of it. Full information of TP, like protein-protein interaction of TP53, and what are the gene-gene interactions of TP53, and how about the motifs, domain regions, and who are the authors who are posted, the sequence, structures, information. Like this, plenty of information you can find in these gene cards. The next database what we see is the cancer proteome atlas. Okay, like a genome sequences. Even if you want to get about the proteome information, one can go with this TCPA portal. Just in Google type TCPA portal. So in this URL, we can able to find this information. And for targets, you want to open any specific targets, okay, for a disease, you can use this website. GEPIA server. So this is to identify the gene expression. If you want to filter the genes based on the expression, okay, like if a patient is suffering from a bladder cancer, okay, what are the genes responsible? So like this specifically, can if you take 1,000 genes are responsible, then what are the top most important genes? Or if you're focusing on one particular hub gene, what are the top 10 genes interacting with the hub gene for the bladder cancer? Like this, we can see all these gene expression patterns through GPIA. For gene enrichment, go with Enrichar server. This is the URL for the Enrichar, myanlab.cloud Enrichar. So go to this server and you can do the enrichment. Like if you want to get top 100 genes, or if you want to go with top 1,000 genes. So all these things we can able to make it out. So what are the top 100 upregulating? What are the top 100 downregulating? So this information we can also enrich. That is, we can do the gene enrichment for a particular target. Okay, type a particular target and go with the enrichment using this. And this is an exclusive database developed by the PG scholars of SVC. Actually, in this database, what we have developed is we have developed an adjuvant database. You know what is adjuvant? Adjuvant is a biomolecule which can, this is used to increase the efficacy of antigen. The antigen is generally used to secrete antibodies to for getting a prolonged response. In the vaccine, we use antigen. So if you want to increase the efficacy of a vaccine, okay, we use biomolecules like adjuvants. The adjuvants can be of protein molecules or it can be also be plant molecules. So earlier we use adjuvants of inorganic type. The most popular adjuvant is the alum. Okay, so like this we have used the metal salts also as an adjuvants. But nowadays we are working on organic adjuvants to not get accumulated in our biological system. So my research focused on plant-based adjuvants. So I have taken 1,000 types of interactions with the plant-based adjuvants. Okay, whatever I interacted with the dendritic cell target proteins, surface proteins, I just deposited through SQL. This is the database language I used, and PHP is the front end language. And also I have used a .NET framework, and the programming majorly I used is the C sharp. So these are the tools I have used, and we have had around 1,000 interactions. Like for example, if one is going to search for rheumatoid arthritis, what is the best plant-based adjuvant we can use for arthritis vaccine? If you're searching for a malaria, what is the best adjuvant we can use for uh, the malarial vaccine? Like this, we can able to find it from this database. So that's it. Now, so something we have a few minutes time. I can take it to the web and I can illustrate you. Come on. Just we have 2 to 3 minutes. I can take you. So can we first go to the Stitch DB? Stitch. Okay, Stitch database. So in Stitch database, stitch.embl.de advanced. Go back to safety. Oh, presently this is having some problem. What problem? Whether I can able to not secure. So now it's going inside. Okay, click search. So 1.6 billion interactions are there. 2031 organization. It looks similar to the string DB, but interactions are less. There we find 20 billion. Here 1.6. Type any specific thing like Thafrin. This is a plant compound. I'm typing the plant compound. Type any drug also and check whether we have drug interactions. We don't have? Not found? I don't know. Can give something indomethacin. So indomethacin continue. So indomethacin interacting with these proteins. We can also increase the number of interactions, and we can also find this kind of a drug protein interactions. Okay, now let me take you to the next database. The next database, what I'm going to show is the FunCoop. FunCoop. So this is also a gene protein association networks. So inside that, give any gene identifier. SARS-CoV-2 is an example. So if I click SARS-CoV-2, so many genes are there. So I click search in the FunCoop. So not only can give a particular gene, you can also give multiple genes. That's what you can able to find in as a keyword in FunCoop. We'll be getting after some time. Let me take you to the next database. I told you C Bioportal. This is a common database for cancer. If you are working on cancer, this is also most important portal. So go to this kind of types of cancer, any type of cancer information you can able to find it here. And here you can able to find some samples, 2,500, 1,000, 10,000, whatever samples we find here are taken from US hospitals. In America, we have different hospitals, isn't it? So In a hospital, from the different patients, they have taken this information. So one can able to go through this pan cancer studies, pediatric cancer, immunogenomic studies. So all this information, so we can get the data sets information in CBio portal. For doing research, one can also go to this. And for the research purpose, those who are working on genes, better go with gene cards. This is also a reputed database. This is a human gene database. Search for any genes, we will be getting the information on this. So this is a keyword we have. So just typing the keyword and searching through that. So we are getting the information of a particular gene here. If you type a disease name, you get plenty of genes. When I search for hepatocellular carcinoma, I got around 8,000 genes. You can get detailed information of that also. Okay, so if I scroll down, see, lot of data is there. Protein information, protein interaction data. So today we'll be practicing everything. Okay, try to find all these details. And I believe that I'm each and every day before starting the session, I'm sending you some important links to practice in WhatsApp group. Okay, so also go to this kind of an URL and practice it. Okay, so before when don't wait until I send you the video. I'm sending the video on the same day, but before that also, if you want to practice that kind of URLs whatever I'm sending it. Okay, so if you didn't enroll in WhatsApp group, enroll in WhatsApp group. So still I'm sending more information inside. Okay, anything else to be discussed? The most important databases I have provided demo. Okay, even to the websites. Something else whatever I have covered in PPT, you also can go to that kind of a website URLs and you try to explore on that information. So this is the FunCoop information for the SARS-CoV-2. Plenty of genes are there, isn't it? It looks very interesting and funny. Okay, so how the SARS-CoV-2 is interacting with the humans. So these are the SARS-CoV-2 genes, and the blue color are the Homo sapiens. And more about this enrichment, we can do the enrichment here itself. Even the enrichment we can do with the string DB. Okay, other than the tools available, enrichment tools available in this databases, we can also do private enrichment through Enricher. I told you there is a exclusive website called Enricher. Through that also we can do the enrichment. Okay, so like this, we have plenty of information on this. Okay, so one can go to this and you can explore the information. So I have completed my today's session. Any doubts, you can ask me at present. Any doubts? If no doubts, thank you very much for the interaction. Okay, so from the day 1 to the day 3, you interacted well. I appreciate your kind of involvement and as well as the enthusiasm to show on interaction. So let us meet on tomorrow for the day 4 session. So thank you, participants. Bye.